Okay, class 11, finding binding sites. Welcome everybody. So let's start with a reflection on the big picture, where we are in this course. Since you are doing something else, thinking about other classes, let's give a context. This is basically the big picture how I see. I think that molecular biologists need their needs and their wants. No, so you need to know what are the questions that you deal with bioinformatics, and what are the tools that exist. No, at minimum, you need to know what are the kind of input files and what are the kind of output that you will get from these tools. In the idea that, as I have seen in practice, you are not necessarily going to do this in person. I know that some of you will like to do it in person. I am absolutely in favor of that. But some of you will not do this, use these tools directly, but you will work in a team. In fact, in real life, what used to happen is that professors have students, or you collaborate with someone else. I mean, in my experience, it's like typically molecular biologists or biologists in general come to talk to me, saying, okay, I want to do this thing. And I also have seen that, especially in Turkey, people hire companies. You know, you get a project, and you will take your DNA and you will send a company to sequence it. And the company will give you the sequence and it will make some standard analysis. And you have to understand what they give you. And you have to probably deal with them, say, okay, please do this and this and this analysis, and you have to kind of ask the correct question to someone who will do it for you. So that is the minimum that I think you should be able to know. You should be able to work with a company or work with someone else who is going to do it. Then, ideally, if you have more time or more interest, you should be able to use the tools. So how to use that tool? And I make a separation here between the tools that are in the web, like the one that we use so far, and the command line tools that typically, not always, but typically work in a Unix server, not in Windows. Most of the development in science in general, not only molecular biology, but most of the development doesn't work in Windows, because Windows is really, in truth, is a very bad platform for doing this kind of development. And it is so much that Microsoft realized it, and internally they use Linux. And in the last version of Windows, they have a Linux subsystem, so you can use Linux inside. We can try to learn that thing one day. Then there is another topic that is in fact the question I think Recep was promoting, or Arda, I don't remember. One of you were proposing to attend a seminar, super interesting. It was essentially about BioPython, which is an extension of Python to read and write bioinformatic files. Let's say you have a FASTA, or you have a gene bank, or you have many of the files, probably all of the files that we see here. BioPython can read and write them, and it is useful. It is useful, and if we had time, I would love to work on that, but we need time. And there is another tool that we could also teach if we had more time. It's called Bioconductor, which is a set of libraries for R that are also doing the same thing. So, well, Python and R are very equivalent in many things. So it's kind of a matter of taste. And finally, the key point to me is, let's say the most scientific question is how to understand the result. That means you get this result, this number, the thing, what is the meaning, deep meaning of this thing. So in this course, we are essentially in this topic. So we started with the finding data online. We discussed about taxonomy, pairwise alignment that include BLAST, multiple alignment, phylogenetic tree. And we are, as you see, in the middle. We need to speak about DNA sequencing and genome assembly. That is probably one class. I mean, no. The truth is, genome assembly can take 2 classes because there are many topics in the genome assembly. But if we are lucky, we do it in 1 class. You need to understand how genomes are assembled. Then we have, when you get the genome, how to find the genes and what are the genes doing. And one particular point in there is annotation of binding sites, which is going to be today's topic. Primer design is something that you probably everybody should know, and maybe you already know some of it. Is well, primer, how you choose the primer for amplifying the region that you like. And ideally, we should also speak about gene expression, microarrays, or RNA-seq, or something like that. So as you see, we are very much in the middle of the thing, and we are only doing the first idea, like what are the questions, what are the tools. We are doing half of the big picture, and still with that we are in half of the time. And to understand the things, well, you know me, I think that you need to have a very basic mathematical tools. I want to show you something, but you read these names. Give me 1 second. This is mathematics. This is a book about mathematics. It's a mathematics encyclopedia. Okay. Over this one, that is kind of theoretical mathematics. Over this, we have applied mathematics. Okay. This was a gift, by the way. This is mathematics, let's say. Of this, we need like 10 pages. Okay. So you don't need to understand all of the big world of mathematics. The part that are interesting for us are 10, 20 pages, no more. So it's not like so much with no, it's just a little thing, very, very practical things. And to be honest, there are things that I learned in my first year of university, except probability. Probability I learned in the 3rd year. But to be honest, the way they teach me in a way that was very complicated and was not very clear, because it was kind of oriented to researching probabilities. And I think I have a better way to teach that. Is kind of simpler or more based on intuition and more used to understand the results. So I believe that everybody that finished high school can understand all these things. So so how they match, for example, for doing the thing that we are doing, it will be great if you understand that this is the mathematical tools that you need. So to understand taxonomy, you need to understand what are relationships. To understand genome assembly, we need to understand networks, graphs. And you see, the more we go, the more we will need probabilities. So my initial plan for this course was to teach, in this part, to teach probabilities, because it's a tool that will allow you to understand the things. Unfortunately, I realized that we don't have time, mainly because next Friday is holiday. It's a new year. So in fact, we have 4 classes from including this one until the end of semester. And we still have to see genome assembly. Let's say that this is 1 class. This is a class of today. Primer design, yeah, we can maybe make in half day. And gene expression is 1 class. And functional test, yeah. So we are kind of tight. We are kind of short in time, and we will not so. So later I will send you. I don't have a quiz like a quiz today, but I have a survey. I want to ask you what do you think should be the emphasis for the next of the semester? What would you like to learn, and think about this? So well, that is essentially the big picture. No, we are here, and I am well. I initially wanted to speak about DNA sequencing today, but I will do it next class, and I'm going to start with binding site. That, as you see, depends on probabilities, but I will try to not use it very much. In fact, we spoke a little bit about this when we spoke about multiple alignment. I give something very quickly and probably was not very clear, so I'm going to try again. Okay. Any questions so far? Okay. So I like to say, you can correct me, that in general, in broadly speaking, there are three kinds of proteins. And I am interested in particular in the one that are connecting to information. There are other cases that you want to study. In fact, I should tell you something. I don't know if but the study, the proteomic, it's in general something hard. I always avoid it because you normally need a lot of computing and a lot of resources to analyze this. Imagine you to find the function or the structure of a protein. If you only have the sequence and you want to predict the function, what essentially people do is to try to calculate all the physical laws between all the atoms. You have like interaction, like what is the name? Electromagnetic forces between atoms, you know, they attract, they reject, and they have the nuclear forces like a weak interaction and strong interaction. That's kind of the basic laws of nature between the 100,000 atoms that you have in a typical protein. And then you are trying to find what is the shape of the protein that will give you the lowest potential energy. That, in short words, is what people do in protein modeling. Now, that problem typically takes hundreds of computers for several months. So I always avoid it. Now, just before you went to the exam, like 1 month ago, there was a company that published a method that do it in half an hour. They found a machine learning system that can solve the protein folding problem faster. So, for example, some researchers say that they were working in a protein for 10 years and didn't get any result. And with the new tool, they found the result in half an hour. So you are replacing 10 years for half an hour. And I know, for example, there were some talks in Izmir, the Izmir biohealth, what's the name? The IBG, you know? They were testing it. They were doing it. They have a good bioinformatics group and they are already testing. So it will be great if we can do that. But my point is this, I have not used it and I don't know if I will use it soon. But you, in your professional life, will live in a world where people can predict the shape of protein. I think many people think, and I agree with them, that this is going to be a game changer. Like the point that you go from 20,000 to 100,000 proteins with a shape that you know, and you can predict or model the interaction, is going to change the biology, the way that we understand molecular biology upside down. I really think so. More computers in your life, in other words, more computers in your life, because you will have more data and less need of a wet lab. Then if you can predict that, you can also predict what are the enzymes or what are the chemical reactions that they can catalyze. That is the second part of knowing the protein. Now, analysis of enzyme is something that we have a separate course, is system biology. System biology mainly deals with enzymes and the effect of transcription factors. So I always think that system biology is the second part of this course. Just to give you an idea where we. Yeah, if we had more time and a good team of, no, this semester I have 0 students in system biology, but if we had a good group, we can try to do something with this thing. Anyway, so yeah, prepare for the world. The world is changing. Well, from all these things, the part that I want to take focus today is transcription factor that I will define for the purpose of this class. You correct me if you think the protein that can bind to DNA. It is true that there are more than transcription factor. I mean, the replication, what is the replication factors, the DNA polymerase, and so they are also binding to DNA. But in general, what I want to give the name transcription factor to any kind of protein that will bind to DNA and do something with it, like start transcription, block transcription, do something with the transcription. And in particular, in fact, I'm not thinking about the protein. I am thinking about the binding site, that is the place in DNA, in the genome, where the proteins bind. These are usually short. I mean, we are speaking about around 20, maybe 15, maybe 25, but it's not 200 base pairs, okay? It's a short segment. And the basic point is that the same protein, or the same family of protein, they can bind in different places in the genome. So you have one protein with several binding sites, and you will like to characterize what they have in common. So for example, I show you some classes ago, I have an example that you can follow in this web link. I have the page, so I will show you in a minute. But with different experiment, ChIP-seq, or you know better than me, you can essentially isolate the protein when it's bind to DNA, and then separate the DNA, and then sequence the DNA. And you know that these are the sequence where the FNR protein binds. Okay, there are more. Let me show you. I have it here in the corresponding here. Product here. So I hope you can see it. If not, let's make it a little bigger. So you have these are the sequence, and if you click, eventually you see what is the paper that supports this thing. Sometimes the sequence is short. I'm not counting them. I'm counting only the ones that are full here. Okay. And here you have a link to the protein. In this website, they have the different name for the object, but you can click and see the protein. And but we're not looking at the protein here, as you see the protein. And we are not on that. We are essentially looking at this set of sequences. Okay. Any question? No? Move. Use microphone or move the head. Okay, if you don't have camera, you have to speak, of course. You move your head without camera. So we want to find a way to characterize all of them. In other words, we want to find the motif. You know, motif is, I think, German, maybe Latin. It's like, what is the idea behind what they have in common? I know. You know, motif, I know the leitmotif is something that people use in music. You use something to say that a song or a piece of music, you have different parts, but there is kind of a part that is repeating. There is a kind of underlying song, a piece that you find it in many parts in the music. That is the motif. I will not sing, but you can imagine. For example, the typical is the Fifth Symphony of Beethoven. The motif is very clear, no? It's kind of famous. So we want to find a way to characterize the protein. There are many ideas behind that, but the one that we're going to see is this. We start counting, and we probably spoke about this. We start by counting how many times you see every letter in every position. So let's go back a minute to the, what is this? Here, no? No, here. So you see most of the sequence start with a T. So you count and you have 27 times you see a T, one time you see a G, one time you see a C, 3 times you see an A. Okay, we're essentially counting how many times each one. Fine. Okay, this is the matrix. I copy the matrix from the website. And I'm calling this matrix F, but I am writing like this. So F, you take two inputs, the row and the column. The row is ACGT, and the column is 1 to 14. And the basic point and the difference with BLAS is that here we consider that the columns are different. In BLAS, the score in every column is the same. Here we have a scoring that depends on the column. A different column, we have different rules. If we have more time, we can compare this with BLAS. So you have this frequency table, and for example, A1 is 3, C1 is 27, and C6 is 7, no? This one. Okay, everybody agree? So, so something that, well, I say what is saying here, but you probably saw it already. If you look at here, you will find that the sum in every column is the same. So you have 27, 30, 32, 32 here, 32. And the sum of the matrix was built with 32 sequences. When they do the multiple alignment, you have 32 pieces of DNA. So there are 32 values for the first column, 32 for the second. Every column has 32 things. That thing can be A, C, G, or T, but these are the things that we have. Okay? In this case, the alignment is without gaps. And in general, people try to use things that don't have gaps. Okay? You try to make this with the part that is, because they are short and essentially you expect the protein to bind to DNA and without any folding. So you don't have a kind of space for having a gap. So the sum of every column is 32. So I want to help you to remember a little mathematical notation. You know, here we are saying for all J, we have the, for all column, J is the column, for all values of J, the sum of all X, A, C, G, T is 32. Okay, the sum, that means essentially for each column, the sum of all rows is 32. Okay? That is what we are saying here. So maybe you would like to see like this. This is saying the same, but it's longer to write. For each J, this symbol is for each or for all. For each. Oh my gosh, my mouse is dying. Where is my mouse? Okay, so so F of the first row, F of the frequency of the first, the frequency of the second, the third, and the fourth, the total is 32. The point here is that to make this something meaningful, you need to kind of normalize it. Like the result should not depend on how many experiments you did. You want to find the truth of the sequence, not the truth of your experiment. So if you do more experiments, you will get more sequences, or depending on many things, depending on the organism, whatever, you will have more and more sequences. So if one binding site, if one transcription factor has only 4 binding sites, you want to divide by 4. If you have 400 binding sites, you want to divide by 400. So in other words, we want to have the relative frequency. Okay, this is what we really care, not the how many times, but what percentage of time you see every letter in every position. So for example, 84% of time, the first letter is a T. 94% of time, the first letter is a T. 94% of time, the third letter is a G, and so on. Okay, well, this goes to the side, but you follow the idea, okay? Therefore, the new matrix essentially for every row, for every column, the small f is the relative is essentially the big F, the one that we counted, divided by the total. Okay, maybe I should write it in a nicer way. Okay.
Okay.
In this case, the total are all the same. There is a question, no? No. Okay. But in general, you can think that the column may have different totals. And obviously, in the relative matrix, the sum of every column is 1. That is here. If you do it manually, it may not work because you may have some kind of rounding error. Like for example, here you have 84 plus 3 plus 3 is 90, and this is 99. There is some rounding error here in the presentation because I'm using a small number of decimal. But if you use all the decimal, you will find that this is 1. Okay, every column is a total of 1. Okay, because that is the definition of a relative frequency, is the percentage of every case, and the total percentage is always 100%. Okay, it is very simple. I'm just saying it hard to scare you. Okay, now I'm going to scare you. Then we build a matrix that we will call the position-specific scoring matrix. That is defined as this. It is, we will call it M, also row 1 column. X is the nucleotide, J is the column, and it will be the number 2 plus the logarithm of the relative frequency, the logarithm base 2. Now, the question that I hope you ask is why? And the answer is, aha, you should take my probability class. It's a longer story, so but I will give you an intuition, okay? Question?
Why 2? Is it the...
Why 2? Well, you should take the probability class. But I will give you an intuition. So the result is this. Before going, I want to show you something. First, when the frequency was 0, absolute and relative are 0 in both cases, obviously. And the logarithm of 0 is minus infinite. So that is kind of very ugly, no? Then most of the numbers are negative, but some of them are positive. And if you see, the positive are the ones that have higher frequency. Sometimes 1.6, 1.7, 1.7. Why? Maybe, can we say, no, there is, well, okay, let's see how we get. So let's see the intuition that let's see if I can. First thing first, since the frequency, the relative frequency are between 0 and 1, they are always, I mean, relative frequencies are always less or equal than 1 by definition. It's a percentage, okay? A percentage of a total. So it can never be higher than 1. Now, if you remember your high school, logarithm of 1 is 0, and logarithm of numbers below 1 are always negative. I like to say that in biology, biology in general, the only function that you need to really know well is logarithm. It's the basic thing. Logarithm is something that you should know very well, so you don't get surprised with things. I should make a list of exercises, so you practice until this seems come natural to you. So if the number is less than 1, then the logarithm is less than 0. The logarithm is always negative.
But still, in this case, they use 2. I mean,
I'm going to. So why the number 2? If all the sequences has 25% of probability, they will have the same chance of 25%. The function, the logarithm of the 25% will be log 2 of 1/4, which is minus 2. Here, this is minus 2. Okay. So this will be, so if you have 25% of probability, you will have minus 2. Let me, yeah, this is a minus, okay? I will fix it in a minute. Therefore, if I add 2 to this logarithm, you will get 0. If a nucleotide, every nucleotide that has 25% of probability, or 25% relative frequency, if you like, this log will be -2, but adding 2, it will give you 0. So why we care? Because this is the basic point is this. If the frequency is more than 25%, in other words, the nucleotide is more frequent than you can expect just by random, then the logarithm +2 is positive. If the nucleotide frequency is less than 1/4, in other words, if the nucleotide appears less often than you will expect by random, then this number is negative. Okay? That is the relation of number 2. The number 2 comes essentially to what we are doing is comparing with 25%. So the assumption that we are making here is that in general, all nucleotides appear 25%. So the summary is this. Score will be positive for the nucleotides that are frequent. Frequent mean more than 25%, and negative for the ones that are less frequent, meaning less than 25%. Okay?
25 just relies on the, we have 4 letters, right? So each of them has 25%.
Well, it is not obvious. I will go to that. I clicked too much. Yeah, what happened here? 1 second. I want to show you the, okay, here. This is a formula. And so again, the high frequency, if the frequency is more than 25%, we can see here, this is more than 25%, this is also more than 25%. So in general, when they are more than 25%, not in general, always, the more than 25%, you will get a positive number. And if they are less than 25%, so what I'm doing here is this. I am making the assumption, as Ahmed said, I'm making the assumption that all nucleotides have the same probability, a priori. Okay? And I compare the intuition of the probability, the a priori probability, with the real values in this case, and we find that some are more often than we expect, and some are less often. So this is the bottom line. The score is positive for the nucleotide that are highly represented, overabundant, if you like, and is negative for the ones that are less abundant. Okay? Question? No? No. Point. As you say, we assume that the background frequency for each nucleotide is 25%. That may not be true, because you may be working in an organism that has a GC content, let's say 30%, 35%. If the GC content is 35%, then you will have G or C, let's say 15, 17% of C, 17% of G, and I don't know what is corresponds, 30% of A, 35% of T, something like that. Okay? So you may need to correct the formula, and the formula can be corrected. The program that uses this thing can make this correction. But for today, we are going to assume that we don't know the GC content, and therefore we're assuming that all the nucleotides have the same chance. Another practical issue is that if the numbers are 0, the score is -infinite, and that is kind of very pessimistic, essentially, because if you get 1, 0, you will get all -infinite, and -infinite will kill everybody. It's a super killer. So that is not a good idea, and what we'll do in practice is make this correction. And the logic is this. We did several experiments, but we don't see everything. We only see the experiment that we see. So if we get 0 in a column, it may be because we had bad luck and we never saw the good case. It may be that later, with further research, you find a new case of a binding site where that number is not 0. So there is a small chance that you can play with that small chance, but there is a chance that you are missing something. So one of the ways that people solve this Well, one way is that instead of minus infinite, you just put a number, a negative number, a large negative number, but don't care very much. The other that you will find in many of these cases that you take the absolute number, this 25, 28, and you add a 0.1. That will not change the total very much. And then you of course, you have to normalize, but these corrected numbers. So in this case, the relative frequency will never be zero. In the worst case, you have 0.1 divided by a big number, but not zero. And something else that is, so what else? The first practical issue is that the frequencies may be different given the GC content. You have the right to adapt them. The second issue is that you need to correct for these cases that there should not be really zero. There can be a small number, but not zero. And the third point is that, at least in all computers, integer numbers are much faster. I don't know if that is true always in today's computer, but in general, for let's say physical issues, if you don't count decimals, think how much. I mean, imagine when you multiply two integers, you just multiply them. When you multiply numbers with decimal, you have to multiply them and carry the where you put the comma. If you are moving the decimal point forward or backward, you have to think about that and correct about that. So Atakan put the chicken scary with yeah. Ahmed has another like people scary with math. Don't be scared. Well, okay, I will scare you. Do you know what is this symbol? The kind of half bracket?
We use for concentration usually, but
no, but the concentration is the bracket in both sides. No.
oh, this is a different one. I thought
This is only half.
Okay.
This is called floor. Floor. It means you take the integer part. Floor essentially is like if you have a decimal, you go to the integer that is just below. So 1.5 you get to 1. 1.9 you get to 1. It's not rounding. It's well, you can also round, but I just wanted to show that there is a symbol for drop the decimals and take the number that come before. Since the number are small, I multiply by 10 before. Why is this? Because later we will see that you remember the PAM matrices and the Blossom matrices, they are essentially doing with the same thing. They are also a logarithm of something, and then they multiply by 10 or by 2 or something like that. They multiply by a constant, and then they take the integer. If you remember the matrices that we saw when we spoke about BLAST, they all were matrices with integer numbers. And the reason is because it was much more efficient, probably is still more efficient working with integers. At least they use less memory. And the result is not necessarily 100% exact, but almost always the same. So you really have to be very unlucky to have a case that the result is not the same. And the good ones are going to be good and the bad are going to be bad anyway. And in fact, this rounding error is less important than all the hypotheses that we made before. We make a lot of hypotheses. I mean, if I have to criticize this model, I have 10 things to criticize before this rounding. The rounding is not the critical point. But at the end, people will use matrix either with decimal or not decimal. For the class, I'm going to use the one with decimal because it's easier to explain, but you can see that in practice, people use matrices like this with positive and negative numbers. Positive are good things, mean that they are very frequent, and negative are bad things. So how we score things? First, just for a small case, let's see that you have a small sequence that I call W because I'm thinking of a word, okay? Just for the same size as the motif. So here, for example, we have 14 columns. This motif has 14 columns, And then let's take 14, a set of 14 letters. Maybe we'll be able to do it in Excel. If we have time at the end of the class, we do a practical thing in Excel, okay? Let me go to the slide first, and we do the practical before the break. Tamam? So essentially the score is very easy to calculate. You got this matrix, and you take for every column, you take the, let me explain here. For example, let's say that you have something that starts with ACGT. Okay, maybe it's not, no, something TCGA, no? TCGA. So the first column has a T, and that gives you 17 points. Then in the second you have C, and they give you minus 100. So you take 17 minus 1,000. Then you have G in the second column, in the third column, TCG, and A in the other. So you get 17 plus 19 plus 17 minus 1,000. That will be the score. If you have TTGA, you will get 17, 19, 19, 15. So if you choose the good ones, you will get positive numbers. If you choose the negative, I mean, if the sequence has the good letters in the good position, you get a positive number. If the sequence have the bad letters in the position, you will get negative numbers. Okay? So you follow the logic. Okay? It is kind of, the point is that the rule depends on the column. When you move a little, you will have a different rule. But it's a very easy way to calculate a score. You only have to believe me that you take this log and add 2 or something like that. Well, you realize that some columns have very high frequencies and some have, let me go to the matrix here. So for example, here you have 17, 19, but here in the middle, you don't have big numbers. You have 9 is the best number here. You have 5 here. Here you have 15, but in kind of in the center, you have 7. They're not very high numbers in the center. So in the border, you have high number. In the center, you have kind of low numbers. And you would like to see what is the importance of every, I mean, how much can you expect to get from every column? So one way of doing it, and kind of the official way, is that measure how much information you get from every column. We calculate essentially an average, like you have the score and you have the frequency of every nucleotide for the same column. So for example, you know that 1% of the time you get, let's go here. If you remember, we had like 1% of the time, if I not, yeah, there was 2% of the time you get A, 1% of you get C, 1% you get G, and 85% of the time was 17. So you have 17 positive 87% of the time, and I don't know, 1% of time you get this minus 30, 1% is minus 30, and 5% is minus 15. And you calculate that average, and you get a value of that we call information of every column, and that essentially look like this. And I want to stop a second. Well, let's stop a second here. So we have the average of every column. This is a weighted average, no? I mean, the letters that are more frequent count more, and the letters that are less frequent count less. So the first is good. The second and third are very high. The third go down, then in the middle you go down, and then the top go high. Okay? This shape is very typical in binding site. If you see more and more binding site, you will see this and more. And in fact, you probably look something like this. You have seen, this is this called the sequence logo. What do you see here?
So middle parts tend to be more flexible?
Exactly. Middle part tend to be more flexible. Let's take a minute to first thing. So do you recognize the shape of this drawing? No, the drawing here, the size of the letter is proportional to the frequency of the letter. T is the most frequent. T is the most frequent in the second. G is the most frequent in the third. A is the most frequent in the fourth. Okay. And if you go back to the matrix, you will remember that the first. Shall I go back, or do you remember? The first letters were T, T, G, A, T. Okay, the most frequent. So you see here what are the most frequent. But some of columns are higher than others. Why? Because of this. We're essentially following this thing. Okay, maybe I have something a little different here, but you see that there is taller and shorter later. And yes,
may I
go ahead.
ask? So the sequence we use W is not part of our experiment, right?
No, it's not part of experiment. It's like how I will test a new sequence. How will I score a new sequence?
Okay.
It's something new. Yeah. In fact, the logic that I'm trying to say is this. First, I give you a rule to calculate this M matrix, the matrix for scoring. Then I tell you how to apply. If you get a sequence, that is the score that you will apply. Then the score, as you see, depends. You get one score for every column. You get a score in the first column plus the score on the second column. You go from the first to the last column, and you got something from the first, something from the second. So my question is, well, what is the average that you expect for every column? In every column, you expect essentially a little from the first, a little from the second, and at the end, you will get that the column that give you more on average are the first and the last, and in the middle they don't get you very much. And that is also reflected in this. There are tools, and in fact, if you Google sequence logo, you will find tools. This I did in R, by the way. There is a tool in R to do this. But I want you to think about this. So essentially, what we are saying is that the T in the first is important. The T in the second is super important. The G is super important. In the middle, I don't care very much. And then at the end, I care.
So bigger letters represent conservative regions. So
Exactly. The more conservative. You have to be careful with the word conservative, but yeah, your intuition is correct. Conservative, because conservative normally is you see in the sense of evolution. Here we are not speaking about evolution. We are speaking about different sequences in the same genome. But yeah, the intuition of conservative is the correct one. It's a measure of conservation, if you like. Well, I say level of conservation. I say it wrong. Yeah. Yeah, people say conservative, but we have to be careful. That's kind of the more high frequency or more dominant, if you like. What else you see? Biology's question.
It's like palindromic.
Exactly. It is palindromic. No, that is not obvious a priori. If you look at the sequences, they are not necessarily palindromic because not always start with T, not always end with A. But on average, they are palindromic. So you have TT with AA. That is not necessarily the case, but happens to be. So therefore, they probably have some biological reason for that. Now, I am also, of course, not an expert on this, but I understand that the one kind of transcription factors have something called zinc fingers that bind to DNA, like they have two parts, and they bind to DNA in two parts, and they need to find in one strand you have TTGA, and in the other part, the other strand is also TTGA. No, coffee, Roman, coffee. Remember that the DNA has 2 strands. Now then, other biological question, why the center is not important?
There may be some other proteins that.
Yeah, there may be, but in general, if you look at transcription factors, binding sites, you will tend to see this. This is very, very common. No intuition, no idea? Well, some part of DNA are important and some part of DNA are not yet so important. No? Well, the theory is this. DNA is folding. So there are some part of DNA that are exposed to the protein, and then it goes down and is not exposed to the protein, and then it goes up. So essentially, the DNA gives a turn, if I'm not wrong, every 26 nucleotide, but the half turn is 13. So between here and here, you have like half of DNA turn. This is the part that exposed to the protein, then it folds and the protein cannot see what is happening there. So if you change any letter, the protein doesn't care. And then it folds back, and this is the second part that the protein will see. And in fact, in this part is looking at the opposite strand. So what is seen in reality is TTGAT in the opposite strand. So you essentially are looking at an effect of the DNA folding, of the DNA 3D structure.
It's good.
It is good because
I like it.
it is something that we only took sequence, we only took letter, we count frequency, and we do some analysis, and we find deeper structures. So,
I was thinking more like animational way when DNA transcribes, like they open into lateral, I mean, two strands, and I didn't think about turns at all.
Yeah. That is the point. That, I mean, someone told me. I didn't think about this. Someone told me this. I read somewhere, but my point is that like we did with the PAM matrices, we in the PAM matrices again we only took sequences. We didn't think anything about chemistry, and the rest will say chemistry. The rest will say these 2 amino acids, given their conservation, that was evolutionary conservation, they have similar properties. And that is something that you can see in the sequence. That is the magic of this thing, that you get letters. You forget everything about the chemistry and you only get letters. And when you do things with the letters, you find truth about chemistry, truth about the structure of the DNA and things like that. I even had an idea that if you look carefully, you may be able to find part of DNA that are folded in a different way. That was a project that we discussed some years ago. Okay, how we use this thing, a little application. Any question before we go further? I mean, the point that I want to emphasize is that we do this looking for insight, looking to understand better what is happening. That is the magic of this thing, that with the good analysis, we find things that are not obvious a priori. Find the main insight. So the logic is more or less obvious, and I think I will go to the I will go to the Excel now. But let's finish the slide. So now we have a genome, millions of letters. and you would like to see whether, in fact, there is a strategy that instead of looking in the complete genome, you look for binding sites only in the upstream regions, like, well, depending on the problem, of course, but if you are concerned about regulation, the basic assumption is that the regulators will attach to the promoter region. Now, what is the promoter region? That is a question that some people asked me once, and the truth is the promoter region is the promoter region, and there is no good way to characterize. But usually what we take is like from the transcription start site, we take 1,000 or 2,000 nucleotides upstream and maybe 100 or 200 downstream. Okay, so you have a gene, the gene has, I don't know, 3,000 nucleotides. You forget about that. You take only the first 200 nucleotides of the gene, and then the 1,000 that happened before the gene. Okay, that's the upstream region. Now, you need tools to find the upstream region. It's typically not something that you get from NCBI, but Artemis can do it. Artemis has a tool like give me the 100. They give me the upstream region of every feature. Or you can make usually make a little BioPython program. Because, for example, in bacteria, essentially, in bacteria, we think that binding sites are outside the gene. So the upstream is before the start of the gene, but up to the previous gene. In bacteria, genes tend to be very clustered together. So sometimes you don't have enough space. You have like 100 nucleotides or 30 nucleotides between genes. They are very close together. In bacteria, you have operons. So essentially, you expect the regulation to be at the beginning of the operon, not at the beginning of the gene. So you need to identify operon and so on. In eukarya, of course, the things are different. You have a lot of space between genes, and you have a lot of space, and then the upstream region is probably longer. The promoter region is probably longer. You take 2,000, 3,000 nucleotides. So, but the truth is, nobody knows really how long you should look. So, well, the other option is looking in all the genome and then see what is close to a gene and what is not. And then test. Okay, all these things in the computer, you have to test them experimentally later. So how we calculate? Well, the formula, you see it here in the screen. You get the genome. The score depends on the genome. Different genome will have different score. And then you have I, the position inside the genome. And what are we doing essentially? I put N, I should put L here. Essentially, what we have is you start in a position, you take the 14 letters and you get the score. Then you move one and you calculate a new score. You go one by one, calculate a new score. So this is a window of a fixed size. The size is the size of the motif, 14 in this case. So you take the first 14 letters, you calculate the score. Then you move one nucleotide and you calculate a new score for the next 14. And then you move one, and then you move one. Okay? And you go like that. You follow me? And then you calculate the score and you will get what are the points with higher score. Now, there is a story that it will take much longer to explain. That you need to calculate some statistical significance. But the tools that we use do it for you. If you want to know the statistical significance, take the probability class. So, for example, I will suggest this thing. The tool that I use for this, maybe you can find better, but the tool that I use is called the meme suite. This one. And here you have the complete sequence. So, for example, the two parts. One is like, given the DNA, how you find the motifs, and the other is like, given that you know the motifs, you have a database of motifs, how you scan them, motif scanning. So here are the tools. The one that I usually is this FIMO. And you should see there are different different things. So you take your sequence, you take your database of motifs, and you look for them. So, well, I'm not suggesting that we do this right now because you have to finish the exam, but this is the tool that I wanted to show you, and if we can make an exercise later. I don't know, maybe you can try something here. What does this happen? Motif databases, we have new thing, blah, blah, blah. Okay, later. They are working on this. That is at least good. This is a project still in development. So, okay, good thing. You can upload your motif or use some. Okay. We need to find the motif, and I don't have it at hand. So let's do this later. There is a format for this, and yeah, we should speak about the format. Like
I was just gonna ask that we have a matrix. What's the format for?
This. There is another project called Jasper when they create these matrices. I took mine from Prodoric, but Prodoric is, for example, a project that has not been updated for 10 years, so I'm not sure it's the best. Jasper, and the sad thing is this is the kind of tools that are useful for medicine, for pharmaceutical. So many of these databases sooner or later become paid. They are not for academia, for pharmaceutical companies. Pharmaceutical companies have millions of euros. So these are usually paid data. So here you see the matrix here, log odds. That is the technical name of the matrix that I show you, positive and negative numbers. Here you have 4 columns for A, C, G, T in this order. You have, this is the valid letters, 4 columns. And the alphabet length is 4. The width is 15, if you have 15. And I don't know what is this, E. Here you have the frequency. This is the total number of sequences that they used to make this thing. And this is the name of the motif, you see. And then you have another one. Name and the log and the frequency. And the URL where you can find the original. So it's looking like this. Well, maybe we can use some vertebrates. Of course, you need. Well, I didn't say this thing, but the real hard part is this. With these tools, we can find binding sites, but it's not always easy to know what was the protein binding to that binding site. The relationship between transcription factor and their binding site is not obvious a priori. Where is the class? I lost the class. Here. So I will do something. I will prepare an exercise, and I will send you some tools. But let me prepare with the proper set of data. Okay. But we essentially need 2 things. Okay, let's look at it again. We essentially need 2 things. We need the database, the motif database that look like the one that they give you, and the sequences that you want to look. You can upload your sequences or use something like upstream sequence of plants. for example. I don't know. Then you can find different. Well, they have some organisms that are already prepared. Prokaryotic. You can put your email because it can take some time, so you get the result by email and a description so you remember. And you cannot give more than 80 megabytes. Okay, this is still working. Okay, so here in prokaryotic you have different prokarya, bacteria, let's say, and you may have different versions. So if you it's kind of, you give your own motifs and then you look for these motifs in different organisms. In my case, I was doing the contrary. I was taking standard motifs that I get from Jasper or somewhere like that, and then I was applying to my bacteria that was not public. Eventually, you can download this software and download and install. And run in your computer. You can, yeah. You can run in Windows with simulation of Linux. Okay. No, but we can use it on the web, so it will be good enough for a practical. But I prefer doing something in Excel. Okay. Let's do this thing. So the question, I showed you this slide before, so I'm going to go through them very quickly. How to find the motifs initially, if you like, in this pipeline, like this part, the motif discovery. Okay, how these things work. So let's do it quickly. If we know the matrix, the position specific score matrix, this M is technical name is the position specific score matrix, because it has different score for different position. It is easy to find. I just say that you go through the genome, go through the sequence, calculate the score, and the highest score are the site that you like. High score will be the new motif. So then you can do it aligning, but if you don't want to align, that was a thing that we talked before. We essentially do this: given n sequences of let's say the same length m, and a motif width that here I call k. Maybe I should call k anytime. So we look for a k-mer, a subsequence of k that is overrepresented. And we are essentially trying to see which k-mer is more frequent. Let me give you the context again. Let's say that in a microarray experiment, you get 100 genes that are reacting to the same condition. You have some input, and you have the cells normal and cell with a different condition, and you have 100 let's say that you put some compound, you put something in the medium, and the cells react in a way. And with the microarray analysis, you can identify 100 genes that are reacting together. And the hypothesis is that these 100 genes that are reacting together are essentially regulated by the same transcription factor. And you would like to know what is the binding site for that transcription factor. So you have 100 genes, you took the upstream region of these 100 genes, and you want to see in this upstream region what they have in common. You have, let's say, 1,000 letters in front of 100 genes, and you want to find a letter of size, let's say 20. What is conserved? What are the 20 letters that are conserved through all this 1,000 sequence? So K in the example I gave is 20, and you want to look through all of them. So here I say with text, as I show, but let's go to the graphical representation that I'm saying the same that like in the text. This is a method called give sampling, and it's very crazy because it's very random, but it still works. So these are the upstream region in light blue. These are the region where you are going to find the motifs. Then you have no idea. So whatever you choose will be fine. You choose something randomly, and this will be very bad, of course. But it's enough to you get your data. You build the matrix. You build the scoring matrix. You do everything. You separate them. You get. They will be very different. So it is noisy at the beginning, but at the end it will allow you to calculate a score in different parts. And the higher the score, this one will give you the position of the better new element. So you go replacing one by one with the new sequence that was the better score. And then you get a new matrix and you improve it. So you start with a very bad matrix, but that matrix allows you to find sequences that will make the matrix better. And you repeat this thing. You choose one sequence and you use the matrix. You build the matrix with all the other sequences. And you look in the sequence that you choose. Let's say that you choose the second one. So you build a matrix with the first, the third, the fourth, and the fifth. With this one, you find the sequences, you find the matrix, and you look in the second one, which is the best matching this one. And then you put that back in the list. That is essentially the idea. You start with something random, you repeat, and until the system doesn't change anymore. The magic thing is that it eventually finishes. It eventually cannot improve anymore. Now, if you are lucky, you find something good. If you are not lucky, you don't find it. You get something not so good because there is a lot of randomness here. If you see the detail here, we are choosing randomly the sequence and we are choosing randomly the starting point. There are many random. So what people do is that you do it several times. And the reason why we do randomly is essentially because we don't want to be trapped in a bad decision. Like you choose randomly and that can be good or bad. So if you do it again, you want to do it differently. So since we don't have any information a priori, we just do it randomly. And the good thing is that at the end, it works. It tends to work in most cases, of course, not 100% sure, but a very high probability. So we have some tools. In fact, this is the old motif. This is a Belgian group, University of Ghent. If you want to know, University of Ghent had a good. I mean, I used to have a friend that worked there, so I have good opinion of them. I mean, I still have the friend, but she doesn't work in Ghent anymore. So the give motif sampler is the original idea. You can look here. And the second is this motif suite and meme and must. So we have this suite doing this strategy with some variations. You have the motif suite in University of Ghent in Belgium, and this Brown University. Let's see what happened. So in summary, we have two questions that are kind of chicken and egg. One is, given the sequences, how you find the motif, and the other, given the motif, how you find them in the sequence. So it's a little chicken and egg, but interestingly, random works here. This is not working. Randomness works here. You take an initial point and you get something. Well, that's it. So before we get too crazy here, let's do an exercise. I have, what time is it? Let's copy this. Let's see if I can copy to Google Sheets.
I want to, the last few slides were a little bit unclear for me. How do we improve the metrics? Maybe before the break.
Okay, well, you have to read them carefully. I went both times that I show them very quickly. But the way that we improve the matrix is this: you choose subsequence. You choose words initially, and you make the matrix. Then you use the matrix to look for new sequences again, and so the new sequence will get into the matrix. You drop the old one and you put a new sequence. The matrix, this matrix, remember, corresponds to a set of sequences. There are 32 sequences here represented in this matrix. Okay.
Okay.
So, and I can use this matrix to find new sequences like this one. And so when I find that new sequences, I can improve the matrix essentially, including new sequences that match the matrix.
How about the initial matrix then?
That is the point, that the initial matrix is essentially random.
Oh.
You start with one subsequence for every upstream randomly, and you will get a matrix that will be garbage. Okay. But even if it's garbage, you can use it to find new sequences. And the new sequences will match the garbage. And then, since it's garbage, you take one that is good and you throw the old one and put the new one. And you repeat again.
Okay, and I see now.
That is how it's going to improve. You start with the garbage. You use the garbage to look in one of the sequences. And you find in the same sequence one subsequent that is better matching the matrix than the original one. So you drop the old sequence from the matrix and you put the new one. And you do it again and again. You start with random position and then you choose randomly one of the sequence and you improve it and then you choose. And eventually you cannot improve it anymore.
Because we have a limited sequence, right?
No, because essentially you get to a point that when the matrix is kind of stable, you use the matrix to look into the sequence and you will find the same that you had before.
So it doesn't make a difference.
It doesn't make any difference. So that is when the process stops. When you try to improve and you cannot improve. When the matrix is not giving you any new sequence, you don't have any way to improve it. When that happens, well, you cannot improve better. The only option in that case will be start again, and that's what people do. You do it once, you get some result, then you do it again, you get another result. Of course, this can be done much more smart. There are smarter ways, and the tools that I'm showing you do it in a more efficient way. But it's just to give you an idea how this thing works. The key idea that you have to keep in mind is that the process happening behind is has some randomness on it. It's not 100% sure. There is some variability on it. And that is something that you are going to find more and more. That is kind of, I don't know, I forgot the name, maybe stochastic method. But the basic point is that in biology,
heuristic.
Yeah, but the heuristic is more general. You can have heuristic that are not this kind. For example, BLAST has not random on it. BLAST uses index to simplify. This kind of heuristic are stochastic in the sense that, I mean, the philosophy is this: the number of combinations that you have to explore is so big that you cannot explore them all. So you choose them randomly. And if everything goes well, choosing randomly will be good enough. It's the same like when you do an interview, a survey, or you instead of asking everybody in Turkey what is your weight, you take samples of people randomly, and that random sample will more or less correspond to the total of the country with high probability. When you, or let's be more clear, when you grow plants in the lab to make an experiment, you are testing only, I don't know, 10 plants, 100 plants, nothing more. And you are trying to tell the truth about all the plants. All experiments that we do are sampling because you cannot test all living organisms. You can only test one particular, and you want to generalize from this particular case to the other. Okay, I think we are running out of time for our break, but just to finish the idea here, an exercise that we can make with a little more time is from here, make the second and third matrix and then test some sequences. So essentially you will do something like, okay, an easy way will be to copy this here and here, and then just to have the shape, and here I will change the formula. So this is going to be equal to this divided by the sum. Well, in this case, I know that it's 32, so I'm going to be lazy. Not the best strategy, but we are short of time. If you want, we can make it a little bigger so we can see the decimal, but it's more or less what you see. And then the score matrix will be, no, let's make something here. Let's make this be 2 plus 0.1. Let's do the correction here. We put a parenthesis and divided by 32.4. 0.1 4 times. Okay, that will get rid of the zeros. And we reduce the number of decimals, so okay, not too much, something like this. 10%, 3%, 84%. And then we calculate the formula. We say 2 plus log, do we have, no, log of this in base 2. And let's reduce the, yeah, so the exercise will be something like this. And finally, you have to calculate the score. You have a sequence, I don't know. Okay, I should make big letters. A, T, G, A, T, G, C, T, A. All right, big letters. Damn it. W. No, not W. Not T, T, A, whatever. Okay, I have to make it uppercase. How we make it uppercase? Upper. No. That is just because I'm lazy to write with pressing shift. And then essentially you have to look. Okay. Do you know this function, the lookup? No, there is no, like, what is the name? What is it? Index? No, what is match? This is the modern way, match. Match of this letter in this range, no? I think it is. So I is the first. Wrong. This has to be $. You know how to use Excel, no?
Not this.
You don't know dollar, or you don't know match? Match is new, so.
Match is new, yeah. You talked about dollar before, I think.
Yeah, I think. So then you have something called index. Index, and here you have a matrix, and then you say the row and the column. So this is the matrix is going to be this, and I have to put dollar here, but and the row is going to be this number, and the column is going to be this number here. Wow, move away. This one. Ah, maldición. No, we have to put the dollars in the correct thing. The matrix will not change, so we put dollar everywhere. Here the 21 should not change, and then the because the column will change, and this also the same. I think that will work. So that is the score for here. A 1.4, 1.9, 1.9, 1.6. You can verify it, no? And then finally, some of this. Yeah, some of these things. Score 0.2. Now, how can improve? Let's say instead of a T here, we have an A. The score improves, no? So, and the point is that when you get a sequence, we should get a sequence. When you get a genome, you are taking the 14 letters, the first 14 letters here, and then the second and the second, and so you're essentially doing the same operation that we are doing here through all the genome and getting different score. And the higher score are the ones that look more probably like a binding site. Well, I am out of time now. Timers say that we are out of time. So break time, okay?